Okay, here we go. 10, 9, 8, 7, 6, 5, 4, 3, 2, 1. Good afternoon, this is Dr. John Bennett broadcasting from Miami Beach, home of neurosurgical. 7. Oh, hold on, I made a mistake. I gotta start again. Hold on. Hold on, I'm sorry. Good afternoon, this is Dr. John Bennett broadcasting from Miami Beach, home of neurosurgical. Oh, that's bad. Hold on, I made a mistake. I gotta start again. Hold on. Hold on. I'm sorry. Good afternoon, this is Dr. John Bennett broadcasting from Miami Beach, home of neurosurgical. Oh, that's bad. Hold on, I made a mistake. I gotta start again. Hold on. I'm sorry. Good afternoon. That's what happens when I put YouTube on at the same time.
Okay, your voice was like,
Yeah, okay, it's off now. Okay, we'll start again. 10, 9, 8, 7, 6, 5, 4, 3, 2, 1. Hi, this is John Bennett broadcasting from Miami Beach, home of Neurosurgical TV. We have the pleasure of having the 4th episode of an embryology series from Neuro-Health Care for LMIC. It's a channel that's going to be regular. We're trying to determine if we're going to have it twice a month or whatever. Anyway, this is a series of 4 on embryology, and I'd like to introduce the host and the presenter. Atta Bhatti is a neurosurgeon, originally from Pakistan, now in Switzerland, and he's training to be a dual neurosurgeon. So we appreciate that, and then welcome, Atta. It's all yours.
okay. Can you see my screen?
Yes, perfect.
Okay, good. Okay, so today, in continuity of our neuroembryology series, we are trying to understand the basic neuro development in human. And today we have selected metameric segmental basis of CNS development. This is a concept which is very fascinating. And in the current era, when the spectacular progress in developmental neurobiology, the tremendous advances in neurogenetics and higher resolution of the modern imaging techniques, this developmental process of human brain and spinal cord have been much more progressive. And after 100 years momentum, with what people learn without and these technologies is now having more concrete possibilities in human. So just to quote that, this is Dan Donkelar, Martin Lemon's clinical neuroembryology, which besides other papers and neuroangiography and neuroembryology, other references, we followed this for our basic presentation. So we know that the neurons are natural migrants. So they are the cells which are responsible for building up the CNS. Most, if not all, the neurons in the mammalian nervous system, they migrated from a place near the occipital and neck region in adult. In growing embryo, that's where a lot of activities comes with the second weeks of development in the embryonic period. In the brain, neurons usually originate in the ventricular zone, what we call as subventricular zone, where their precursor cells proliferate. They can then migrate in a defined destination. This migration is radially to other layers and tangentially and very precise. So this process leads to the coherent um, union of different pathways leading to the formation of different neuronal structures, including the cranial nerve. Neurovascular system also works in parallel. There are many temporal and many stimulus in the face of genetic programming and chemicals which govern, and most of them are initially produced by the placenta and then neural tube notochord itself. So here in this, we see that the neuronal migration is a crucial step in the neural development, as defects in neuronal migration causes multiple human diseases. So we try to explain that the nerves, CNS, and different components like the prosencephalon, mesencephalon, metencephalon, and rhombencephalon, um, myelencephalon, as well as the whole spinal cord. They have a segmental distribution, and different segments joins during the developing embryo. And as we see that in this picture, the neural tube formation has happened. We see the endodermal yellow layer. So this is the stage when the non-neural and neural tissues from the neural crest cells, they uh, migrate, and this is the 21st stage, 26th day, 21 somite stage, 26th day of life, and you see a different components eventually. Also, the mouse is little faster in the growth at that stage, and there are 39 somites in the mouse embryo. But we see the color scheme showing the cranial nerves area on the dorsal side of the head side of the embryo, what we call the cephalic side, the dorsocephalic. The cardiac system is cardiac beats, and cardiac system is advanced. Sacral and spinal cord somites, these are there. So we see the notochord formation. As we know that the mesodermal derived notochord gives a lot of chemicals which helps in guiding those cells to proliferate, to migrate, to go elsewhere at their target areas. And this precise ability of embryonic, this neural crest drive cells is an amazing capability. So it's very time bound phase. Within the uh, second to in the end of the second week of embryo growth, the after the fertilization, implantation happens in 1 week, eighth day, or the seventh, eighth days. So initially the fertilization, and then there comes to the proliferation of cells from the 2 cell to 4 cell morula, and then blast stage. And we see that how this process goes. The to set the stage for the activities, there are co-regulators, there are genetic information within the cells. The pair rules, there are genes, there are many names even skipped, hairy, expressed, prefigure. We'll see it in next slides. But these neural crest cells, they are the pluripotent cells. They have ability to form any tissue, any future tissue, whether it's cartilage, bone, nerve, neuronal tissue. So this pluripotent potential cells is something which has capability and has sense of time-bound activities and the coordination. And after the formation, from the neural crest, these formation of mesodermal cells of pluripotential cells transform into stem cells. They crawl through elsewhere. This is called as migration. And this embryological distant area, they invade, they join, they become the part of the new cells, new structures, and new functions. Origin of neural crest cells, as I mentioned, multipotent, delaminate from the origin and migrate throughout the body to differentiate into several cell types, including cells of peripheral nervous system, melanocytes, cranial cartilage, bone. So the facial, head and neck, these are the close allies, and cranial nerves and the other muscles of the head and neck and intracranial structures, including the dura, blood-brain barrier, and these all are eventually being the product of these cellular activities. In humans, at least 47 cell types have been defined as neural crest derivatives. So proper neural neural crest cells migration relies on environmental cues, and there are ephrins. These are described by different authors. Semaphorins 3F, versin, chemokines in the form of strong cell, and many others. So here we see the anterior pore is still open, which is the cephalic side of the embryo. Posterior neuropore is visible, the future cloacal membrane, rectal, and the genital areas. We see this thing happening at the end of the 4th week. A week prior, this embryonic flat surface was starting to show the notochord and the midline ectodermal tissue. It invaginate into groove-like structures we call neural groove formed from the neural plate, and this makes the neural crest formation and the somites on the lateral sides. There are lateral tissues, lateral mesoderms, and spinal cord, white matter, central canal, somite, and spinal cord structures. They have a 4th week appearance in an initial phase. So we see that the neural tube derivatives list of structures like ventral horn cells, preganglionic autonomic nerves, the astrocytes, oligodendrocytes, ependymal cells, retinal cells, posterior pituitary, cortical neurons, gray nuclei of brain, and the spinal cord. They are neural tube derivatives. And the neural crest derivatives are cranial ganglia, dorsal root ganglia, autonomic ganglia, Schwann cells, adrenal medulla, and melanocytes. So this process of migration, in these slides, we see that there are the possibilities of anomalies at the neural migration and the organization. In the brain, we see the germinal zone. This is third ventricle. This is the subventricular zone. And we see the relationship of wall of the lateral ventricle and the germinal matrix to the developing cortical plate in a harmonious, very organized way. This cells formation starts from the subventricular zone. It's a very highly organized process which leads to the um, help with the help of these radial fibers. These neurons, they form, they go to the cortical layers, they make connections, and here we see the glial cells making the radial fiber path, which provides the migration process. And this is the anomalies we see during the 7th week of intrauterine life, a proliferation of neurons, as I mentioned, that in the subependymal walls of the lateral ventricle, we see the germinal matrix is the term used for it, and then proliferation of these neurons eventually leads to the formation of cerebral cortex. There comes a period of radial migration, and this at the 8th or 7th to 8th week of gestation, the appearance of brain becomes more human-like, more organized. Sulci gyri appears, and the rest of 8th week plus life is having more sulci gyri, and before that, all the structures, the basic structures, make it look like a human brain. This migration of neurons is facilitated, as we saw, the radial glial fibers, and we these fibers, they extend from the ventricle surface medially to the PL surface on the cortical edge of the cerebral hemisphere. And the avenues, the connections, the neuronal migration that makes different pathways, fiber tracks, the different layers of the complex. So to give you the idea, how from the 4th week of the gestation to the later months of the pregnancy in a neonate, how the sulci and gyri appears, how the insula becomes more active at the opercular surface. You see the insula is insula, and deeper to that is the areas. So cerebellum and the brainstem is also visible. So we will connect this concept of segmentation, the neuromere form. This has a different models. We are going to discuss it briefly, and we are more accustomed to spinal segmentation, and as we know that there is a metameric basis of spinal development, and there are 35 metameres, and each mere has its own bone in the form of a vertical body, and it's also developed from the neural tube. So I'm just trying to connect to the caudal end of this column, and I will show the dermatome myotome diagram also, which gives a segmental appearance of human cartoon. So we have 35 metameres, and there is a very organized bone, neural element, nerve, root, covering, dura, sclerotomes, muscles, and so each segment has its own um, on components of vertebral body, neural soft tissue elements, and muscles or bone. So this is your architectic organization of nervous system, where every metameric basis, metameric uh, segment or metamere is part of independent unit with its own blood supply, its own nerve supply. So this segment um, uh, combined with the vicinity segments to form the segment, and the similar way the neurovascular development happens at this stage and makes different tracks. So coming back to the cerebral neuromeres. So neuromeres is the segment of the central nervous system, and at different places, like in telencephalon, we call it telomere, the prosencephalon and the rhomboencephalon, we call it rhombomere, mesomere, and we will see how they look like in the growing embryo, the different flexures, which we saw in details in previous lectures. So 20 cerebral neuromeres are present, and as I mentioned, 35 segments of the spinal cord have a metameric segmental structures. These 20 cerebral neuromeres contemplated in the prosomeric model, which is the most recent for advanced 2007, 8, 9, 10 era, when this prosomeric model was more getting popularity in terms of its acceptability in the laboratory, its genetic basis, and the different testing abilities to confirm the cues of um, details in human embryo. So clinicians have been aware for a long time of the segmental significance of objective dermatome and myotome. For example, this S1, L5, we know the L5 has its own, um, I mean, when we see the spinal cord, we see the cervical enlarged element with the cervical segments, then the dorsal with the lateral intercostal nerves, and then at the conus, the lumbar enlargement is seen. And from the lumbar enlargement, we see the conus medullaris and then chorda equina. Although the segmental skin distribution is a different place, it's not right on the top of the L1. It's little lower, and the myotomal and the neuronal mechanism is still far. So this is how the metameric segments of spinal cord are, which we are used to, and this similar process goes on at the CNS level, at the brain level, at the cranial level. So we are trying to just see to review this: the embryonic period, fetal period, perinatal period. The embryonic period is initial 2 weeks of the gestation. From the third, um, uh, there is one embryonic period from the second to the 8th week, and before the implantation, the 2 weeks are the early life, when the morula and blast stage, and before pre-implantation stages. So in the fetal period, which starts from the 8th week, um, we in the embryonic period, we see the formation and separation of germ layers, the neural plate, and heterogeneous cyst and fistula, split notochord syndrome. If there anything goes wrong during the neural plate stage, we see the cyst and fistulas in the midline, split notochord syndromes, etc. Then it comes the dorsal induction time, 3 to 4 weeks of the development, when the neural tube, neural crest, uh, derivatives, closure of rostral and caudal neuropores, and paired LR plates appears. And these are the major morphological events we see. Anything goes wrong here will lead to anencephaly, encephalocele, neural tube defect varieties. Myeloschisis, the most extensive form, myelomangocele, Chiari malformations, etc. So ventral, during the 4 to 6 weeks, the telencephalization or ventral induction. So the ventral part of the embryo starts showing more improvement of the features, like the development of forebrain, face, formation of cerebral vesicles, optic and olfactory play cords. Although they are lateral, but they are more growing and more human-like. And We also see the rhombic limb appearance, fusion of cerebellar plates. This is all going on side by side. There is pharyngeal arches, pharyngeal pouches, simultaneous way the neurovascular development also keeps on going. So, but in the ventral induction is more relevant to telencephalization, and anything goes wrong here leads to the formation of holoprosencephaly, Dandy-Walker malformation, and craniosynostosis. So once we come to the fetal period, the neuronal and glial proliferations until the 16 weeks of the gestation, cellular proliferations in ventricular and some subventricular zones, which I showed in previous slide. Early differentiation of neuroblasts, glioblasts, cellular cellular regression, apoptosis, migration, and these external granular layer of cerebellum formation, Purkinje cell formation. This happens during the fetal period. And any insult due to any reason, the nutritional infection, any other ischemic or insult to the growing human embryo can lead to microcephaly or megalencephaly. And it can be a genetic problem leads to the formation of this microcephaly. We see Zika virus in recent years, which leads to the microencephaly, and the usual cause is mother's viral illness caused by the Zika virus, leading to the impaired growth of that pregnant mother. That pregnant mother acquired the Zika virus, and it leads to the damage to the developing human embryo, leads to the microcephaly. Similarly, megalencephaly also happens at this stage. And we see that during um, the second and third trimester, so these are the weeks after the fetal period, like after the 8 weeks, the parallel things has been shown in this table. After the 8 weeks, there are a lot of leap forward development in the brain of the growing human embryo. And migration keeps going even after the natal life in the postnatal. So there are different connections, synapses formation, and these migration in the internal cortical neurons, corpus callosal and white matter organization. And similarly, we see that the vascular development comes with the growing regional demand, and this neuronal migrational disorders are the features when something goes wrong in the migration and connection processes. And we see the agenesis, lissencephaly, polymicroagenesis of corpus callosum, polymicrogyria, schizencephaly, heterotopia, etc. And in the perinatal period, the advanced human embryo, the major morphological features, late migration, organization and maturation of cerebral cortex, synaptogenesis, and this again, cortical dysplasias are the resultant abnormalities if something goes wrong. Myelination is the process which keeps on going until the 2 years postnatally, and here we see the secondary acquired destructive lesions in the myelinated fibers. So we walk through this table again, classification scheme for malformations of cortical development, going into little more detail of malformation secondary to abnormal neuronal and glial proliferation or apoptosis. These are 4 types: decreased proliferation or increased apoptosis. This is the feature of severe congenital microcephaly, and there are many forms of dysplasias, microlissencephalies, and in increased proliferation and decreased apoptosis will result into megalencephaly. So microencephaly, there are more accelerated apoptosis, and in the reverse direction, megalencephaly is the condition which happens if there is a increased proliferation. And so there are two ways um, in which the neuronal growth happens, by the positive stimuli, when the growth of number of neurons and neuromeres is accelerated, and the other one is regression. So there is a very tight, balanced way in which time-bound regression or acceleration of the growth of these neurons happens. If that process gets disturbed, this leads to microcephaly or megalencephaly. Similarly, cortical dysgenesis with abnormal cell proliferation. These are the part of glial proliferation abnormalities, neuronal and glial part. And second is malformations of neural migration. So just naming it again, with malformations with neuroepithelial abnormalities, they are of various types: anterior predominant, posterior predominant, so involving temporal or trigonal areas, periventricular heterotopia, ribbon-like heterotopias, malformation. So these are all neuronal migration defects. And we can also see the generalized abnormal transmental migration, and it clinically has effects on diffuse classic four-layered Liz and SPH. So these are the, again, X-linked lissencephalies and different different syndromes. We don't have time to go into the details of these individually. It's just giving you the basic concept of how the neuronal migration leads to the clinical presentations. And malformations at the late radial or tangential migration can lead to sublobar dysplasias. Malformation due to abnormal terminal migration can lead to the different congenital muscular dystrophies or cobblestone malformations. So there is a generalized rule that if something goes wrong initially in the development, the effects are more global. As the time advances of the embryo growth, the effect becomes more focal. And there is a slide which can demonstrate this concept later in this presentation. So the third form malformations class is post-migrational development phase, in which the cortical malformations can happen, and we can see the calcified schizencephaly, transmental clefts, and other abnormalities and syndromes like Adams-Oliver, Joubert, and in cortical dysgenesis secondary to metabolic disorders, we can see the mitochondrial pyruvate metabolism and paroxysmal disorders. And in focal cortical dysplasias, we see different type of presentation, cortical malformations. Post-migrational developmental microcephaly can lead to encephalopathies. So morphological segments or neuromeres of the human brain, described by the Barthelmess in 1923, and later on in 1952, Buxquist. Neuromeres are segmentally arranged transverse bulges along the neural tube, particularly evident in the hindbrain. So the process is more visible, more active in the hindbrain, what we call as rhomben cephalon. Of gene expression studies, we have various genes, starting with the home box and expression of Hox genes. And each neuromere has alar and basal components. And in the developing spinal cord and brainstem, sulcal limitans, that's the midline, divides into the proliferative components into alar and basilar plates. The mesencephalic, the midbrain region or mesencephalon, that's the part of sulcus. This is not continuous with a more rostral diencephalic sulcus. So diencephalic has its own segments, which we see in this slide. So this was more than 140 years when the embryologist or described, he was aware of neuromeres and the developing component of the brainstem and diencephalon. Later on, there was a dull area when there was no further interest, but now with the gene targeting has confirmed the existence of striking patterns. The genetics is much more advanced, and now we see a lot of interest by different groups in different fields, including embryology, gynecology, in vitro fertilizations, the stem cells applications, pluripotent cells research areas. So a lot of things are vibrant nowadays. So commencing in 1995, a series of detailed analysis. The Poils is a pure sciences research person, Professor Poile, and his work and his prosomeric model is very popular amongst the researchers. And here we see that the hindbrain has initially been described as 12 segments, and uh, uh, this initially was described in by many groups, and in 1995 Polish work has highlighted it further. These 12 segments called 11 rhombomeres and the isthmus. So there are very defined horizontal zebra-like strips on the brainstem region, rhombocephalic region, where isthmus or separation of the boundaries. So these are the genetic derived rhombomeres. Midbrain has two segments named mesomeres or midbrain posomeres, and they have been labeled as MP1 and 2. And the diencephalon has three segments, DP1 to 3, described by again Poiles et al. Gene expression. He was the keynote speaker in one of our Zurich neurointervention courses, and was invited by Professor Valavanis, who has described these mesomeric neuromeric patterns of AVMs involving the particular neuromere rhombomere, and according to the AVM classification, he was pretty clear that the AVM in the brainstem, for example, or diencephalon, they follow the pattern of these segments, and the future classification would be like the DP1 AVM with feeders naming XYZ and the venous drainage. So this was the embryogenic explanation of the vascular malformation, which was clinically observed in more than 1400, 12 to 1400 AVM cases done at University of Zurich by the series, presented with some of the greatest successful stories in the AVM all over the world. So coming back to hypothalamo telencephalic prosomeres, HP1, HP2 are again this are being described. So a lot of repetition to understand the same concept. So here we see the neural tube, the sketch of neural tube. The midline is visible. Here we see the neuromeres from mesencephalic M1, M2 to R0, the boundary between the mesencephalon and rhombencephalon, and somatomeres on the lateral side, and the pharyngeal arches. So the somites, occipital somites are also visible, and the arches 1, 2, 3, 4, 5, 6 pharyngeal arches are also visible on the more lateral side. So segmental arrangement of the developing head and neck in the form of neural tube is divided into two mesomeres of the mesencephalon, as I showed, M1, M2. Rhombomeres, initially 8 rhombomeres and then the 12 rhombomeres with the growing embryo. And Paraaxial mesodermal cells which flanks the neural tube, both neural crest cells and somites contributes to the pharyngeal arches, which are 1, 2, 3, 4, and 6. They provide the basis of all the head and neck structures, including the vascularity, the muscles, cartilage, bone. So the neuromeres and pharyngeal arches uh, appear to be the primary drivers of segmentations in the head. In the similar way, R1 and R3, we see R1 to R3, they contribute to the pharyngeal arch 1. Um, uh, um, and the neural crest form the posterior mesencephalon and upper metencephalon, as well as the fourth and fifth cranial somatomere supply of the first pharyngeal arch here, the somites. So they are interconnected because they have to make a harmonious group of tissues, nerves, vessels, cartilage, muscles, and cranial nerves. That's this head and neck stage, which starts from the pharyngeal arches, the pouches, the clefts, and this area leads to the formation of various neuronal cues and migration of neurons alongside cranial nerves, as well as the tracts and the neuronal components of the cerebral hemisphere and cerebellar hemisphere. Similarly, the neural crest from the mid-myelencephalons, primarily the R6 and seventh cranial somatomere, migrate to the third pharyngeal arch and neural crest from the lower 7 and 8, um, um, also the myelencephalon, and second, fourth occipital somites migrate to the fourth pharyngeal stage. So the interest area of the vascularity in the form of vasculogenesis and angiogenesis has evolving human embryo feature, starting from the primary divisions, the 3-cavity formation, 5-cavity formation, then the metameric stage, territorial um, uh, growth, and leading to the more angiogenesis, vasculogenesis process, supplying the ischemia as a potent factor to recruit the vasculogenesis and angiogenesis. And then the remodeling is a lifelong process. So we see that in the developmental stage, if something happens, it has a more global impact on the body. This is the target tissues. When something goes wrong with the remodeling, which is genetically programmed. So when somebody says, is AVM a genetic disorder? So is it present at the birth? No, it's not necessary, because the same remodeling can announce this AVM at later stage as a focal abnormality, as a lesion, and there are many factors involved besides the genetics. Then hemodynamics comes in, and the neogenesis, angiogenesis processes take shape, and to give various progression in the AVM, we see the nidus progress in those cases where we follow up. So similarly, we see that the defect later on at the metameric stages leads to the dysplastic familial or non-familial diseases. We named a lot of these cortical and neuronal glial abnormalities in our previous slides. So metameric, uni or bilateral, one of the cranio anomalous metameric disease described by the Lustonius in hemispheric or infratentorial or uni or bilateral is linked with the metameric level abnormalities. I will show you one of the slide. And during the growth phase initially, it can lead to if there is a trigger. So this is the concept of trigger. If any trigger happens, any abnormality or trigger leads to the possibility of lesion, uh, more generalized in the initial, but the phenotypic expression is not necessarily in every family member or in the same individual. You may or may not have the phenotypic presentation of that genotypic disease depending on the trigger or insult. So trigger and timing is important in all that. So embryologist forms the developmental brain models in the form of neuromeric, or initially the two models, the one model was more to deal with the transversal developmental units or vertical neuromeres, where the forebrain, midbrain, and hindbrain, prosomeres, mesomeres, and rhombomeres and myelomeres were in the longitudinal connections. And then longitudinal dorsoventral zone model is another model besides the neuromeric model. In the dorsoventral zonal model, his model is also a second name. We see the floor, basal, alar, and roof plates. These are the human-made development models to explain the different components of these. So this one is the longitudinal model, and the cisterns, the septi, the sulci, hippocampus in different areas are visible in different color schemes. So somatosensory, vasosensory, and special cranial nerves components are also visible. So you see the telencephalic, the flat unfolded embryo, diencephalic, mesencephalic, and rhombocephalic components. And as we discussed, this is what is the metameric horizontal rhombomeres and metameres and telomeres in other. To describe it a little further, this abstract is helpful addressing the functional implications of prosomeric brain models. So this is a viewpoint, and brain and their structural components of the fundamental structural components of the brain and their mutual organizations can be particularly uh, explainable on these models. And they tell us developmental morphogenetic aspects of the embryo. They can guide us different mechanisms. So modern molecularly based prosomeric model by Pole, which postulates a set of 20 transverse posomeres and developmental units that serves to construct all the cerebral parts and particular typology of many different neuronal populations. They represent the different areas: hindbrain, forebrain, and the metameric developmental units. So each neuromere has its own multiple functions. Most aspects can be navigated through different the markers, and influence the neuromeric growth and monitoring. So these diagrams they tell us the Police model, and we see the telencephalic vaginations, mesencephalic, and the boundaries. And what I was mentioning, that the future AVM classification might be every rhombomere has its own specificity and vulnerabilities at the time of growth, and it links to the triggers exposure. And if we see a brainstem AVM, it could explain these boundaries. And it's not like that, even the glioma also follows the same patterns of rhombomeres. And this has been very well explained, and it's beyond the scope of this lecture to go into that detail. But you see these boundaries, they respect the diseases, at least until the disease becomes, for example, the glioma becomes more advanced and breach through these boundaries. But in the initial phases, we can predict these boundaries as an initial resistance to the proliferation of that, and also the origin of the glial or any other pathologies like AVM. So the giving you an insight into the genetic studies and genetic tools, this Hawk's code, rhombomere, this is characterized by the genetic studies, the timing sequence and appearance of neuromeres and their derivatives can be studied. These are either the aborted fetuses or they are like neuromeres of the, or in various studies, the initial animal models like mouse and others. So the neuromeres of the forebrain, midbrain, and hindbrain were determined morphologically in these strains. This again shows the segmentation of the human, and this O'Reilly and Muller has described the subdivisions, and we see the subpallial, we see the diencephalic, we see the alar plate, basal plate of the cerebellum, collicular, different components of different metencephalon, metameres, prosomeres, and telomeres. So this is how this is the ventral aspect of the embryo. This is the dorsal aspect. We see the rhombomeric rhombencephalon, the spinal and spinal cervical spinal segment. Here we see the prosencephalon, telencephalon. So the forebrain prosomeric field, including the secondary prosencephalon. In the red, we see the evaginated telencephalon. In the violet, hypothalamus, diencephalic components. Here we see in the green, the mesencephalic under the control of the OTEX2 transcription factor, which controls the different components of the growth of these neuromeres. And in contrast, the prepontine rhombomeres at the stemmus level, this is the stemmus level, and this shows the areas, the secreted FGF8 controls these areas. So these are the controlling mediators of the growth and migration and construction of different segments, controlled by the different stimuli in the form of OTEX2 or FT2 morphogen. GBX2 transcription factor from R2 to R4, and then the hindbrain or rhombomeres and neck are in a different Hox gene signal based control. Gene regulatory network, it's an exciting area. So based on the genetic regulatory network involved in neural crest contribution to the formation of cranial nerves, ganglia, and this is a very precise, very controlled, and this dynamic process in the embryonic period, and these are found in precise positions along the dorsoventral and also anteroposterior axis of brainstem, including the mesencephalic region and rhombencephalic region. In the embryo stage 30 days, cell signaling pathways also seen here. We see the cranial nerve growth factors and the different color schemes representing different things. I was mentioning this, Lasdonia's work, the CAMS. This is the head and neck relationship. Insult producing underlying lesions would have to develop before the migrations occur, and thus before the 4th week of development. Like there is CAM1, CAM2, CAM3. So you can see somebody with a forehead or orbital abnormality, but in the form of hemangiomas, some skin vascular lesion, discoloration. But that in a child or in a patient can tell you that it's mandatory to have a scanning in these situations to see um, the actual extent of the disease in the pathways of the migration, which in CAMS 1 can involve the prosencephalic and hypothalamic associated pathologies in the form of tumors. And similarly, the CAM2 have prosencephalic features in the brain and diencephalic and chiasmatic involvement. Similarly, and the maxillary involvement. And CAM3 can have mandibular or cerebellar involvement as well as deeper in the rhombencephalic region. AVM, vascular abnormalities, some bony defects, or some associated pathologies. So that mandates the possibility that the simple sporadic brain AVMs have a similarly early triggers during the metameric stage, and during that stage the genetic information has been coded, and if the trigger happens, it can present lately, or it can, if the trigger appears earlier during the earlier phases of the growth, this can be congenital skin lesions, or along with the CAM 1, CAMS 2, or CAMS 3 features. So this gives a very better insights into the congenital nature of cerebral AVMs. And this was described initially in 2001, 97, in 90s, and then later on in 2000 by the Lestonius. Recently, there were um, more revised syndromes based on the different segments involved, leading to the CAMS 4 and 5. And in the previous, you saw that there are also the spinal components also in the form of SAMs also are the possible associated syndromic features connecting the metameres, specific metameres, and this is an exciting area which is going to give a pediatric neurologist different approach to the patient for the diagnosis. Um, I stop here to just invite everyone's comments and discussions, though it's not a straightforward topic. It needs a second review in the form of taking the different subtopics and the more clinical insights. So this was not possible in a single lecture. So I stop here. Thank you so much for the interest and passion.
Hey, Atta, thank you very much for taking the time from your busy schedule to give this presentation. Okay, any comments or questions from the panelists? And if you don't want to come on the screen, just text it and I'll ask Atta.
Thanks very much for Dr. Atta, this very nice presentation and your information up to date. I asking how many gene we can make it to diagnose from the embryology during the pregnancy. To know what happened before the baby, before it birth, before birthing. Can we make any make a swab or make injection to take from embryonic liquid to know if there's anything more from Jane lab? Thank you, doctor.
Did you understand? Okay.
John, do you want to answer this? You want to comment something, John?
No, did you understand his question?
Omar, please correct me if I am not receiving the right question. Are you just mentioning the genetic details before the embryonic phase? Possibility of detecting the abnormalities during the embryonic phase. This is what you asked. Please just
I
repeat.
mean, from amniotic liquid, before the birth.
Yes.
There's any lab test for Jane to know what happened to the baby before his birth from the amniotic cyst liquid?
Yes.
Jane lab.
Yes, this field is, as I mentioned, that this is a very exciting area, and amniocentric synthesis, seeing different markers like human chorionic gonadotropin, the uh, markers for chromosomal abnormalities. These all are possible, and there are possibility of now invasive interventions in a growing fetus, in the if there is anything goes wrong in. So there are a lot of new things are coming, and the genetics is a more real specialty working along the side in future. At least in the higher income countries, in the developed countries, this is becoming more a routine of obstetric care. So before, during the intrauterine life, it's possible to diagnose various things. One thing is by amniocentesis invasive methods. One thing is just the blood test based the markers, and the routine ultrasound surveillance also can pick the retarded growth or any features of abnormality in the initial phases. Right from the somites stages, the ultrasound has a very good follow-up, basic follow-up tool for obstetric human developmental processes. I hope I
Thank you,
have
Professor.
addressed.
Another question. If we know there is disorders of the gene for neurologic, okay? Can we and study for to replace this gene or this striker or trigger to make good, not to make bad? Do you understand me?
You mean in the
No,
possibility
there
of
is
diagnosing
embryo.
embryonic malformation and to correct it? I think it depends. And if there is a massive abnormality in the growth in the early days, so usually that pregnancy in initial pre-implantation um uh, time, a lot of abortions are the ways to take care of those major abnormalities. But it is possible now by the markers, like I mentioned, the human chorionic gonadotropin, the the chromosomal abnormalities, amniocentesis, if needed. Those sort of invasive approaches can diagnose the neural different abnormalities at different stages. And I don't think it's a standard of practice still to intervene for everything, every abnormality. But it is helpful to decision making of continuing the pregnancy or just uh, aborting that, the therapeutic abortion.
Okay, very good. Suresh has a question.
Yes,
Suresh, do you have a question?
yes.
Suresh
See,
is,
Dr. Bhatt.
where are you from, Suresh? You're from India, correct?
Yes, yes, yes. At present, I am in Toronto, actually.
Oh,
Yes,
okay. Okay.
very
I am from India only. Say Dr. Bhatti, I just wanted to have, could you elaborate about the trigger in the AVMs? What time? And I couldn't follow that.
Okay, so this is genetic. When we were referring these segmentation, because our focus was segments. So one of the presentation of various diseases, because we are talking about segments. Segments have every component, uh, neuronal cells and vascular cells. So any insult during that developmental phase can lead to various possibilities, like AVM is one of that, and it's also genetic components which can be there. So it's not necessary that we see that AVM in the early postnatal period, but the uh, but the probability of having it in later in life in the postnatal growth phase is there if that genetic expression to phenotypic presentation becomes, and usually I mean that the AVM segmental presence, like if it's brain stem AVM, there is a neuromeric distribution of that disease to the boundaries, like if it's rhomboencephalic region, the brainstem AVM. So it will follow the developmental migration paths and the boundaries that particular rhombomer has. So what we see is in most of the AVMs, they are genetically predisposed or intrauterine genetic information or insult to the vasculogenesis, leading to new angiogenesis later in the growth phase or postnatal phase, and then the clinical presentation later, maybe in later years. But in CAMS syndrome, which described in the form of three, the CAMS 1, CAMS 2, CAMS 3, in which there are the migration pathway, the tissues involved in this same one neuromeres, they have, since the neural crest cells have the origin and the face is involved, head and neck is involved. So if there is a CAMS 1, you see some skin discoloration, but the clinicians, there is a possibility that there is a second hidden part beneath the tip of the iceberg, is a hypothalamic vascular lesion. So we as a clinician will not ignore that hemangioma until we do a screening of brain to see whether it's just hemangioma or there is a CAMS 1, for example. That's what I was mentioning. Is it?
Oh, yes, it's clear now. Yes.
Okay.
That's true.
So
Thank you.
that makes us a better clinician. If we know on embryology, we would not just see the surface, we go beneath that.
Yes, that's true. Yes.
Total spectrum of possibilities.
It indicates the trigger of something sitting there at the developmental stage, which you have already described. That's what it means.
As we see many AVMs, they can present late in their life. They may have just a seizure as a presentation, or in extreme cases. So if we know that by doing the complete set of diagnostics, then we can have a better follow-ups
That's true.
and give better chances.
Okay, thank you very much. Thanks for clearing that. Yes.
Okay, any more comments and questions before we close? Fitri, do you have any comments or questions? Can you hear me okay, Fitri? The anesthesiologist from Indonesia.
Yes, hello John, hello everybody.
welcome.
Maybe it's enough for me today.
Okay.
Okay,
Hello Atta.
Hello.
thanks for coming. Okay, well, thanks everyone for coming. Thanks for putting it together, and thanks Suresh, Fitri, Omar for joining us and asking questions. We're not sure. Go ahead.
Very nice,
Someone's saying something? Go ahead.
presentation.
Go ahead, Omar.
Hello to all. Very nice presentation from Dr. Atta. And up to this
Sorry, Omar, did not understand you a bit of what you said. That's okay. We'll talk later. Okay, we're not sure if it's going to be next week or not. We're going to talk about it, but we'll see you. We'll keep in touch and watch the website Neurosurgical TV. Okay, thank you.
Thank you.
Just stick around.
Thank you.
Stick around. I'm just
Thank you.
getting off
Thank you.
live.